So the closer, the deeper into history we go, the more complicated it gets. So if we're looking at the lower levels of biological taxonomy, you know, the hierarchy I showed you earlier on, then at that level comparisons, you can even find species that are extinct. They're still alive amongst us. So you can say that these were living around that many million years ago, if that's the geological time you subscribe to. But if we go far into the species, deeper into history, where the species have become extinct and the fossils that they left were too old to extract DNA from it, then it gets more subjective. So there's more noise in the data as we go further back into the history. And the problem is, that's quite common sense, that the more further back you go into the history, the data becomes more noisy. But for some reason, the Darwinists want us to believe that the further back you go is still as conclusive as the way it was in our modern time. And you know, someone who's got there can think that, look, this is not making sense because I need something that will be, I can observe from that period. And fossils is not good enough because they don't come with birth certificate. So fossils don't have a birth certificate attached to it. They're saying that we were born because we're descendants of this species. You need something more, something that can pinpoint the problem here. And they haven't. By the way, even the genetics I'm talking about, I'm going to critique that. Even that's elusive, because there's problems with that as well, and that's going to come up in the next part. Yeah? So I'll just move on now. So on to my second line of evidence against Darwin's common descent, which was comparative anatomy. So this is based on the critique of homology concept, which is associated with comparative anatomy. And this is the second part, the first part and the second evidence, which was used in the book I showed you in the beginning, order circ bias. So they gave 3 lines of evidence which are overwhelming, permits no other conclusion. So the first one, we saw that there is other conclusions that it permits. So now the second one. So looking at this diagram, an evolutionary biologist will look at this differently to the way you probably look at it. You see this and you say, okay, there's similarities, so what? It's not really proving any point. And you know, for evolutionary biologists, there's some striking similarities here. There's a series of bones. You can see humerus radius, for example, in a human arm, that is in the same sequence in the other species. And whether that's the same bone or not is another problem, because that's the way they, comparative anatomists, they define bones. But they've shown it in the diagrams, they'll show it that these are the same bones found in other species, hence homology. Homology means common ancestry for neo-Darwinists. So this is how homology is defined: similarity of structure and position. So definition of homology before Darwin was similarity of structure and position, and we saw that there is similarity of structure and position. And the explanation before Darwin was common archetypal design. This is a designer's common design rather than any common ancestry. Yet, if we look at what's happening after Darwin's time, you have the same definition, but then its explanation has changed. It's inheritance from common ancestor. So here is just a schematic drawing of what I'm saying. So definition has remained the same, but the explanation has changed. So instead of common design, you have common ancestor as explanation after Darwin. So let's look at Darwin's assumption here. You can see the tree of the tree that he has in the illustration he has in Origin of Species. And here you can see Darwin has a species A and its bifurcating hierarchy. So what he's saying from this is common ancestry. So that was Darwin's assumption in homology, yeah? And this is a famous icon of, you know, the fossils of how whales evolved over about 50 million years, and it came from a land-dwelling animal, which is, you know, Indo-Hyas. And from that, the whales have evolved and became the sea creatures. And the way they construct this is based on the bone structure. So you can see those bone structures in the series, and as the bone structures start changing very gradually, so they will align them up, and then they'll present this as evidence. And this is a few years ago, Ohio State University biologist, his name is Tim Burrow. He published a book against creationism known as Evolution and the Myth of Creationism. And what he presented in this book, this is not the illustration from the book. The actual illustration in the book, I didn't have access to that. So this is an illustration of what he was saying. So he presented Corvette cars and showed that they are descending with modification. So he says, if you look at the 1953 Corvette and compare it to the latest model, only the most general resemblances are evident. So this is the point about the DNA comparison studies that I mentioned earlier on. The general similarities will be seen between the distant relatives, and the more smaller similarities will be seen between the close relatives. So then he goes on to making this point about the 1953 and 1954 Corvette, and the 1954, 1955 Corvette, and so forth. And then he ends the sentence by saying, the descent in modification is overwhelmingly obvious. So you're following his argument, yeah? But the problem is, what's obvious is that this is not Darwinian evolution. This is, construction by design. There were a bunch of engineers who teamed up and constructed these cars. But Darwin needs to show a mechanism that, so what Darwinian evolution needs to show is dust hit the car, or there was wind, or the car rusted and then it went into different shapes. Some kind of natural process that led to the cars changing shape. If you involve engineers, then you are accepting a designer's intervention. And that just destroys his argument. So the critics of Darwinism, they refer to this as Barre's blunder. So he was trying to use this as evidence against the creationists, but he actually reversed back on him, because he's showing you constructed cars by design, by an engineering company, and then that's now an evidence for evolution. Because for Darwin's evolution, similarity is not enough. You need to be able to show a natural mechanism that's also working without an input from a designer, right? So he showed the similarity, but he's missed the more important point. So that similarity is an assumption in Darwinian theory anyway. It's a fact that you have to embrace. But the mechanism that leads to that fact, that model, the car, from the different models of the car doesn't prove that. So what's the mechanism they actually show that to prove this point? Genetics. So neo-Darwinists are gene-centered evolutionists. So they will say genetics is our answer for how that natural mechanism of how one organism goes from one anatomical structure to another anatomical structure is genetics. So they would say that the embryos, the development of embryos is controlled by genes, and those genes then have a phenotypic effect on the way those embryos look like in terms of appearances. So that's the line of reasoning. Well, it turns out to be that that's a big problem for Darwinism as well, because in some cases you have dissimilar genes that are involved in development of homologous features. When I say homologous, I mean features that look alike. So the genes are not the same, but they're contributing to features that are the same. So if you're going to base your argument that the genes are that gene is the natural mechanism underlining these changes, but the genes are also having different genes. So how are you able to construct trees based on genes? Because genes are giving you a whole host of different structures. That's one argument. And perhaps more strikingly and more common is the similar genes. So you've got homologous genes, same genes are developing non-homologous features. Now that's very common in nature. I did my master's degree in transfusion, hematology and transfusion science, but about a third of that was molecular science genetics. So I took a module which is on developmental genetics. And almost every gene that's involved in there, quite a significant amount of genes, they can produce dissimilar structures just from one gene, and vice versa, one structure from very different genes. So that correlation is not there, which they're trying to prove from here, that these structures can be correlated to the genes. So here is a limb patterning gene called distalless gene. Now this, you can see I've pointed arrows at different types of structures it produces. So the only argument I want to make here is this gene is homologous. It's the same gene, distalless, but it produces phylogenetically dissimilar. So morphologically, it produces non-homologous features. So that's one argument. And on the tree that I've been showing you, which is the phylogenetic trees, the species that have those genes are distantly related. So there's a problem for Darwinian evolution here, who are gene-centered, because if they're going to base the argument that these fossils are being constructed in terms of the evolutionary history based on the structure, but the structures are being contributed to by different genes, or the same gene to different structures. So how are you correlating your argument to the genes?
Would you say that genes produce these five forms, or would you say that in these five forms?
So there's two things. In the five varying forms, you'll find the same gene, and that's the more common scenario. And the less common scenario is that same gene, so the different genes contribute to the same limb patterning. So you've got different limb patternings, one gene contributes to all of them in different species.
I guess what I'm saying is it the same thing to say that one single gene is found in five forms, a common gene, but can you also say that that gene has it basically is an effective cause for these forms to be the way they are?
You can say that, but that would be like a common design argument, because if you're saying it's effective cause, but you're going across to different, so you're the branches on the phylogenetic trees. If you got a branch far away that's got the same gene as the branch on the far left, why have they got the same gene if the evolutionary model that Darwinists are proposing is correct? Because it's a common design. It's a common design because that gene works optimally for that particular morphological feature that is in nature. It doesn't argue the case for the phylogenetic tree they are arguing for. They're saying that these genes bring the phylogenetic trees more close, they're more close related, so that's why they got similar genes. Here you got genes across different phyla. So the gene I'm talking about here, you can find in arthropods, you can find in vertebrates, you can find in annelids, you can find echinoderms. You know those phyla I showed you earlier on, and they are distantly related, they're not closely related. So why are you finding the same gene in distantly related that's producing limb patterning of the same kind or different kinds? So that's the argument here. So the argument is that it's not correlating, it's not tightly fitting to the argument that the genetics is the natural mechanism by which these phylogenetic trees can be objectively drawn into the history. Does that make sense? So we have another gene here, which is almost identical, that produces eye patterning. And you can see the fly eye on the left, you can see squid's eye in the middle, and you can see frog's eye. Now these are not morphologically similar patternings. They're different. And they are phylogenetically different. So they're being located in different branches on the phylogenetic trees that the Darwinists construct. Yet the gene that produces these phenotypes or these outer characteristics is the same gene. Yeah? So this problem that I just told you about, this disparity between genes and the morphological features, traits, that there's a big disparity between the two, and then the phylogenetic trees that are constructed on the basis of this. Gavin de Beer, he had this to say: what mechanism can it be that results in the production of homologous organs, the same patterns, in spite of their not being controlled by the same gene? And then he says, I asked this question in 1938, and it has not yet been answered in 1971. This is becoming worse because developmental biology is now growing, and they're finding more genes that are controlling the same features, or more dissimilar genes and more similar genes controlling dissimilar patterning, and dissimilar genes controlling the same patterning. So there's no correlation here. So he goes on to say, it is now clear that the pride with which it was assumed that the inheritance of homologous structures from a common ancestor explained homology was misplaced, for such inheritance cannot be ascribed to identity of genes. So the mechanism that is pinned down to genetics has no base that is objective. So what do neo-Darwinists do? In the absence of evidence, modern neo-Darwinists, they couldn't explain homology. So similarity in structure and shape cannot be explained through the mechanism that was based on genetics. So they do this intellectual stunt. They introduce common ancestry into the definition and the explanation. So here's a guy who proposed the definition of common ancestry. He said in 1859, this is Ernst Mayr, he's one of the famous neo-Darwinists from the 20th century. I think he is. He lived about 101 years, so just yeah, so I think the age has had its effect on him. So in 1859, he said, after 1859, there has been only one definition of homologous that makes biological sense. Attributes of two organisms are homologous when they are derived from an equivalent characteristic of the common ancestor. So what he's done here, and I'll show you this is what he's done. He's redefined homology as due to common descent. So homology is a similarity in structure and position. So he said is common descent, and the evidence is also common descent. So I summarized the problem here. I said features that derive from common ancestry are evidence for common ancestry. So you're always going to have common ancestry in your argument. There's a circular reasoning, you can see, yeah? So here I'm just illustrating that. Here you got Darwin, who was a bit more honest about this problem. He said homology for Darwin was structural similarity, from which you infer common ancestry, right? And at the bottom you got modern neo-Darwinists. They define homology by common ancestry, from which they infer homology. So it's a circular reasoning. So you've introduced within the definition itself the evidence that you are trying to investigate. So here's someone who pointed this out. By making our explanation into a definition of the condition to be explained, we express not scientific hypothesis but belief. But this is a belief that you just already accepted the common ancestry as your hypothesis that is overwhelmingly proven that permits no other conclusion. And then he goes on to saying, we are so convinced that our explanation is true that we no longer see any need to distinguish it from the situation we are trying to explain. So both the definition and the explanation has the common ancestry in it. Yeah, I've got it. Okay, I was saying that we're moving on to our third line of evidence, which is to do with embryology. And embryology is Darwin's strongest line of evidence. And in a letter to Asa Gray in 1860, he expressed his, overwhelming. He was overwhelmed by the embryology and expressed why this is the strongest line of evidence. And he says embryology to me is by far the strongest single class of facts in favor of change of forms. And I'll show you why he believed that. The leading facts in embryology which are second to none in importance. This is in his book. So it's a recurrent theme. And then here he explains why. The embryos of the most distinct species belong to the same class, are closely dissimilar, but become, when fully developed, widely dissimilar. So the argument here is that when the embryos, in the same class of animals, the embryos will be more similar. And the distant class of animals, the embryos will be different. So there's two things, by the way, when it comes to embryological evidence. One is that embryos look alike in the way they form in the process of embryogenesis from beginning to end. So that shows common ancestry for neo-Darwinists, because as the development process starts, they look very similar, and as the time progresses, they start looking dissimilar. So that's the first thing they say. The second thing is recapitulation. So there are two different ideas. Recapitulation is that wherever in your own species you see in the changes in embryos is exactly what's happening in the evolutionary history of that species. So they're two different ideas, but they're part of the same evidence for neo-Darwinists. So neo-Darwinists believe in both of them. So here's the drawings of 8 classes of vertebrate embryos attributed to Ernst Haeckel. These are drawings that are from 1890s, whereas Ernst Haeckel may have drawn this much earlier. And if you look at the top row here, you will notice straight away that there is striking similarities between all these 8 classes of vertebrates. They look more or less identical, or they're portrayed in such a way that they look more or less identical. And this was then powerful evidence for neo-Darwinists that we share common ancestry with all these different classes of vertebrates. But the problem is, in 1998, one of the evolutionary embryologists, his name was Richardson, Michael Richardson. He debunked the idea of the embryology being evidence for evolution by publishing these photographs. Now you can see the photographs at the top. If you go back to Haeckel's embryos, they look similar right at the top, the first stage. But he took photographs of the stages in different classes of animals, and he showed that they look strikingly different. So it turned out that Haeckel actually faked his drawings. And this is well known, by the way. It's well known. Haeckel faked the drawings, and neo-Darwinists took it. And by the way, it's basically, you know, the way we interpret Bukhari, that if Imam Bukhari had said something, we'll go in lengths to explain, you know, what Imam Bukhari meant by that, and maybe Imam Bukhari never meant all of that. So because Darwin has taken this as an evidence, neo-Darwinists will just hold it by their teeth. They will just not let go of it, even after the publications have come out that this was based on the fraudulent drawings. So here you can see the 5 classes of embryos on top, which Haeckel faked, and at the bottom you can see the real evidence. Look at the striking differences. They don't look alike. And by the way, Haeckel, he chose salamander, and salamander just happened to look a bit more similar to the other vertebrates. If you choose frog in place of salamander, it looks far different. And I'll show you in a minute the frog as well. Frog doesn't look like the other vertebrates. 1998. So these are past gastrulation, just past the gastrulation stage, I think just over a month. So that's another problem as well. What Haeckel is portraying as the first stage is actually a stage that comes well after the, you know, the conception, fertilization, and also you got gastrulation and, you know, cleavage. So there are stages that embryo goes through. So he chose the first, which was where the embryos actually converge. They come into similarity with each other, and I'll show you that in a minute. So this is what Mike Richardson said. It looks like it's turning out to be one of the most famous fakes in biology. And he said this in 1997, just a year before he published those pictures, which I showed you. Now this is Stephen Jay Gould. Stephen Jay Gould he's a neo-Darwinist, by the way. He's the one who postulated the theory of punctuated equilibrium. When, you know, Darwin believed in gradualism, things change over time. But fossil record wasn't fitting into that. So they had to explain how come we're getting rapid bursts of evolution followed by stasis, a period of stasis, very gradual change. So he introduced that theory called punctuated equilibrium. So he's a well-known scientist of Harvard University, and he wasn't a creationist, and he was critical of creationists. So he's not agreeing with the religious people on this theory. He's got this to say. But we do, I think, have the right to be both astonished and ashamed by the century of mindless recycling that has led to the persistence of these drawings in a large number, if not a majority, of modern textbooks. And by the way, these drawings are still there in the textbooks. I've got one at home, the drawings are there, exactly the same one. For Tuyuma, and they teach this stuff, but some honest scientists will put a disclaimer that these drawings are exaggerated. But they won't say they're fake and distorted, don't use it at all. They won't say, they still accept this evidence till today. So here's the guy who used the textbook, example in his textbook from 1998. I've got the earlier edition to this, which has this as well. But this is the later edition, 1998. The pictures are there showing Haeckel's embryos. And he actually apparently claimed that he didn't know about it. He didn't know about this problem. You know, then he still persistently used it even after that. So there's a long story behind that as well. Here's another book by Kenneth Miller. Miller is actually a Christian. He's a theistic evolutionist. So he's critical of intelligent design proponents. And he defends Darwinian evolution wholly, entirely, that it's correct, it's the way God has intervened in nature. So he's quite well known for his criticisms against the intelligent design movement. Levine, I don't know much about him, but he's a quite popular author of textbooks and publications. But Miller is quite well known amongst the intelligent design. He debates with the intelligent design movement as well regarding the intelligent design, what's the identity of intelligent design and things like that. He's a cell biologist by his education, and he published this book on biology in 2000. And in there, they've stuck in another bunch of pictures, and basically they want you to believe that there is common ancestry there. So they've already made their mind up. Now you can see fish looks nothing like chicken and pig and human. This is the picture from the book, but that is from page 283. Now somehow, because Darwin's theory expects that there is a common ancestry, so you got to believe that there is some similarity there. So at least they were honest, because they didn't reproduce Haeckel's dubious drawings, but they followed the same concept. And they chose the vertebrates that happen to look alike. And I'll show you how. So here you got what happens before the stage Haeckel has chosen. So Haeckel chose a stage, first stage, which is in a blue box. But if you go before the Haeckel stage, you go cleavage, where division of cells occurs in early embryos. Then you got gastrulation, which is where the blaster folds inwards to make the shape of the fetus. You can see the striking differences there. So everything before Haeckel's stage was strikingly different, so it's been obliterated because it doesn't fit the theory. So remember, the argument was that earlier stages are similar. That's the argument. For common ancestry, the argument has to be the earlier stages are similar, and then gradually there is a dissimilarity as the embryos start reaching the stage towards birth. But here we've got earlier stages that look far more dissimilar than the one Haeckel has chosen as the first stage. So what the developmental biologists call this is a developmental hourglass. So you have earlier stages that look very different, and then it converges into similarities which Haeckel chooses as first stage to prove common ancestry, and then they become significantly different as they reach birth. So they've gone for that middle, where the convergence is, and taken those, and even in the selection, they've been biased. I'll show you. Look at these two embryos, chick and human. They look quite similar, right? And the other amphibian and reptile and the fish don't look like the chick and human. So the chick and human happen to look similar compared to the others. Lo and behold, you see in a textbook, chick and human example being produced because of the similarity. So you're being subjective in your selection of evidence. So if you're going to give evidence of those embryos that don't look similar, and explain that as well. And a vast majority of them don't look similar. And the ones that look similar, they will stick them into a textbook, and student reads it, and student can't see the others. So student only sees what's being told in the stories, just so stories that are being told in textbooks. And they will believe in this. Here's another textbook, Odysseus Bides, which is the one that made the claim in the first place. And they want you to believe that turtle and mouse looks very like the human. I cannot see the similarity here. Maybe I'm not an embryologist to know that, and maybe embryologists see this differently. But embryologists are coming out against this, saying that there is no similarity, or the similarity is very minimal, and it just happened to be similar because species have to go through some similar stages to develop. So it's not a big deal. How did this become the the prime line of evidence here? If they've been programmed well enough during the 3-4 years of the undergraduate study, and this happens to have come in their post, because in undergraduate study this doesn't come, because most science degrees do not teach evolution in undergraduate level. Biochemistry, genetics, and molecular science, biomedical sciences, even medicine, any subject which is related to science, they don't really encounter Darwinism as a subject. They'll just touch on it very briefly, but it's not taught at the extensive level that we're dealing with here. This is at postgraduate level. That book that I showed you, Evolutionary Biology for Humor, is a postgraduate textbook. You can use that undergraduate, but it's not going to benefit because it's too complex. The ideas in there are going into a lot of equations and mathematics. The population genetics is full of mathematical theory. It's got less empirical sciences to it, more mathematics. Basically, they've got mathematician Fisher and Haldane from India and one from UK to get together to develop a modern synthesis that mathematized Darwin's theory. Darwin was wrong about his genetics. He had an idea known as pangenesis. So they excluded that and replaced it with Mendelian genetics, and then they mathematized that and that became the modern synthesis of evolution. So it's a mathematical theory. So if you really go into the detail, you're going to get numbers. And when students read into these numbers, they become programmed to see similarities. So then when they see these pictures, say, oh, hold on, there were some numbers there that made sense, and this must be true. But someone like who's not seen it before, first glance at it, you will see no similarity at all. So here's one scientist who had this to say: only by semantic tricks and subjective selection of evidence, by bending the facts of nature, that one can argue that earlier stages of vertebrate embryos are more similar than their adults. So these are semantic tricks. I'll let you have a read of the summary. And I presented 3 lines of reasoning. One was from comparative anatomy, one was from fossil record, and the third one was from embryology. And you've seen how all 3 of these lines of evidence rest on some claims which you can have a different lines of thought on that. So whereas neo-Darwinists will present these cases in such a way that the student is made to believe that this is fact and there's no second opinion about it. So you've seen that that doesn't stand up to that. Now what I'm going to do, so actually this is just the overall summary. Just give you a couple of seconds to read through the overall summary. So what I've done is I've included summary at the end of each slide, each evidence, so that if you don't remember everything, if you just learn the summary, you will understand what the argument was. And as is expected, short lecture like this, I can't cover every detail. So there is more detail to this. Neo-Darwinists do have their arguments as well, and they will present them. But the point is, these core arguments have not been answered. Even with their new manifestations, these core arguments still stand till today. There's a book written by Stephen Meyer he's a Cambridge, his name is a philosopher of biology from Cambridge University, and he published a book called Darwin's Doubt. That's about the Cambrian explosions. Highly recommended book. And he shows how this is still a problem till today, that they can't explain pre-Cambrian. And you can find, you'll find neo-Darwinists will give you some counter arguments, but they don't really work, because the core problem is still there. If the drawings don't look the way they need to look, the modern pictures we've seen from Michael Richardson that there's no similarity, and we can also we've also seen the homology concept being redefined hasn't solved the problem. That's made a problem go from science into logic, because you're fiddling around with the concepts rather than doing science. So now we're going to move on to the second part of my lecture, which is to do with natural selection. This is what Odisek Byers said is controversial. There is possibility that there's other mechanisms. But the neo-Darwinists, like Richard Dawkins, and you got Jerry Coyne and their colleagues, they hold natural selection to be more powerful than what other scientists will hold natural selection to be. So that's what we're going to critique here, that how much can natural selection do? Is it that powerful then it can create all these diversity around us? It can add information to the genome, a beneficial information or not? So now I'm moving on to the first evidence that used for this theory is peppered moths. So in UK, before the Industrial Revolution, there were more white moths on the trees than the black moths. And then during 200 years ago, when the Industrial Revolution started, the black moths started to increase in frequency and the white moths started decreasing because of the action of natural selection. So the theory was that dark moths were better camouflaged on soot darkened tree trunks, while light moths were selectively preyed on by birds. So what we have here is not natural selection that Darwin needs. This is natural selection. It's a good example of natural selection in that sense that there is, changing frequencies of black and white moths. But this is negative selection. This is purging selection. So whatever's there that's not beneficial purges that away and leaves behind what's going to continue living because it's going to survive. Natural selection, that Darwinian natural selection needs positive selection, which it needs to create new information. It needs to create new traits. Here we haven't created new traits. The black moths were there before and the black moths are there after. It's called natural negative selection. But they usually present this as natural selection in action. So the experiment was done by Bernard Catterwell in 1950s, and this was, and he released some moths onto nearby tree trunks and he started observing what happens with them. And the result he found was birds selectively preyed on white moths, and as a result, dark moths increased in number, which I just explained to you. So his conclusion was this is adaptation to the color of tree trunk. So this is adaptation. Natural selection is working in nature to get rid of the white moths, and the black moths are now well adapted to tree trunk color because the soot that was the kind of powdery deposit that was generated during pollution was being deposited onto the trees, and that caused the black moths to be camouflaged and white were conspicuous to the birds so they could eat them all. And now the problem with that was when he
this is the first time in several months this has happened. It's really strange.
It's okay. Sure. So the problem was that the theory was based on camouflaging effect, but when the pollution was reduced, the white moths increased without the shift in the color of tree trunks. So tree trunks were supposed to also change in color before the white moths come back. But here you go, white moths coming back, increasing in frequency, when the trees are still covered with soot. They're covered with that powdery deposit that causes the blackness. So why does white moths come back? So there's a problem in the experiment. There's a problem in the theory, the hypothesis that this is a camouflage, it's an adaptation to the black, but the trees are not still black, you got white moths increasing in frequency. So there's something else causing this. Another problem was identified in 1980s. So researchers, they discovered that pepper moths don't really rest on the tree trunks in the wild anyway. So Catterwell's experiments were done where he put pepper moths on the tree trunks. But the researchers and there was one of the Cyril Clark and his colleagues, and there was many others, who showed that the peppered moths do not rest on tree trunks, they rest on the upper branches, hidden away in the shadow. So it's not natural for the peppered moths to be found where they're going to be eaten away by preying birds. Whereas Catterwell's experiments were done on the tree trunks. So where did the textbooks get their pictures from? Because these were pictures found in textbooks where peppered moths were sitting on tree trunks. But where are these pictures coming from? So it turns out that this was staged, and they were glued to the dead moths, were glued to the trees, and photos were taken. And this wasn't fraud, by the way, because the theory was there to prove it for them. So it's basically they're getting photographers to take photographs of peppered moths on the tree trunks just to show the students that that's what it is. So it's not fraud, but the problem is it was based on a dubious theory that didn't really stand up to the evidence that was being shown to students. So here's Jerry Coyne, an ardent atheist, and I told you he's a guru of genetics for Richard Dawkins. He says the prize horse in our stable of example has been the evolution of industrial melanism. That was called industrial melanism because, when the dark pigmentation, increases, so it was the actual example is not industrial melanism. So he goes, in peppered moth, this classical example is in bad shape, and while not yet ready for the glue factory, needs serious attention. So he's actually referring to the problem where they're sticking dead peppered moths to the trunks of trees. So he was embarrassed because he's been teaching this to his students for years before he discovered that this example it's not true. And he goes on to say, my own reaction resembles the dismay attending my discovery at the age of 6, that it was my father, not Santa, who bought the presents on Christmas Day. So he was he was surprised that, I'll be using this example to show students, look, natural selection in action, but these are dead moths on the tree trunks that's just been replicated. So Kettlewell's experiments did happen, and he done the experiment during daytime. Moths, they go for the closest surface at daytime. But in the natural habitat, moths don't stay at that surface. They'll go for the upper branches. So there was a flaw in his experiment. So it's not that he did a fraud as well. His experiments were there, but then birds are eating the white moths because they are being exposed to them at the lower branches. So as the researchers, they did research on how moths behave, their behavior and ecology. They found that the moths don't rest that part of the tree, not the lower trunk, at the higher branches. And that's not where the birds have very good access to prey on them.
In this case, it was actually a design element. He was designing the white moths to be placed to eat.
So he didn't design in that way, because what he is he did the experiment daytime. So it's the effect of the daylight. So he didn't, it probably didn't occur to him, Yeah.
It was essentially
Yeah. So and this reveals one fundamental point here, that when scientists do experiments, there are assumptions. And if you don't really know, there's unknowns and there are knowns. So that's why you got to have reproducibility and replicability. Reproducible in different experimental conditions, and you can repeat them in the same experimental conditions. So that's why they have to reproduce these experiments over and over again to prove the point. But if there's some assumption underlying the experiment which was fundamentally wrong, then you're always going to get a wrong answer, though there's the results are coming out to be reproducible. So this is going into philosophy of science now, not science, because in science, scientists sometimes overlook that. By the way, most scientists will come across very arrogant about this. They won't engage with the philosophy of science. They will just tell you, this is the data, and we're more interested in data and what data is showing us. History and philosophy of science will give you a different story. And philosophers tend to be a bit more honest about this than the scientists regarding evidence. Scientists, they stick to whatever they see as evidence that's going to stand forever. Philosophers see the history of it, and they say that, this is likely to change. Like, Thomas Kuhn, his paradigm after him, like that, you got Paul Feyerabend, and you got Larry Lorden, and many others who question these things, which these scientists won't, because they have a commitment to science. The science gives us, truth-like statements about the world. So this is what it was. And yet students are still being taught this idea of peppered moth till today. So here's that, the same two scientists who I showed you before, Ken Miller and Levine. It was logical to conclude that when soot darkened the tree trunks in the area, natural selection caused the dark-colored moth to become more common. Today, Kettlewell's work is considered to be a classical demonstration of natural selection in action. So they continued with this, although this was discovered in 1980s, and we're looking at 2000 now. Still, this was continued. Has it gone off the? So this is just a summary to that one. I won't spend too much time on this. So second critique to second example, Darwin's finches. So here are Darwin's finches, which are found in the Galapagos Island on the coast of Ecuador. And you can see there's about 15 of them with a diverse beak shapes. And they found one island, Oceanic Island. And these are two scientists from Princeton University who did research on one type of species with medium ground finches back in 1970s. And their study was very carefully done. So they actually monitored these birds carefully and what they ate, their lineages. And so they had very thorough study on these birds. So this is actually a good example of natural selection. Compared to the peppered moth, this one's actually a good example. So with a And a good stroke of fortune for the grants, in 1977, there was a severe drought on Daphne Major, which is one of the Galapagos Islands. And this drought cut most of the food supply on the Daphne Major, which is the island we're dealing with. And it killed 85% of the birds in the population. So the birds that it killed were medium ground finches. And this led to approximately 5% of increase in the beak size in a space of about 1 year, which you can see here. This is the evidence we're dealing with, 5% increase in the beak size. So just a slight rise. And the theory was that small beak sizes were less fitter because they were preferentially killed, and the large beaks were more fitter because they were better at cracking the few hard seeds that were available on Daphne Major, and that permitted them to survive the drought. So the survivors had 5% larger than average beaks. So what did the neo-Darwinists do? They extrapolated a speciation event from the initial 5% increase in the beak size. So this part here where you can see the dashed lines is an extrapolation from the actual evidence, which was only a 5% increase in the beak size. So Darwin's estimate was that a drought, if it were to occur in every 10 years, could produce a new species of finch in only approximately 200 years. So every 10 years, if we had a drought, this extrapolation will lead to a speciation event in the beak shapes of these birds on Galapagos Islands. So the problem is that when the drought had ended, the average beak sizes, they returned to normal. So this you can see. So there was a slight increase, 5% increase in the beginning. Darwinists made an extrapolation upwards towards the, you can see the amount of change. This is the amount of change we're dealing with towards a speciation event. But when the drought ended, there was an oscillation, a swing back and forth just about the mean, about the average beak size that should have been the sizes of the beaks originally. So we have a beak sizes oscillating here about the normal value ever since the drought. So the actual evidence is what you can see in this line at the bottom, and this is extrapolated evidence. So there's no net evolutionary change. And very few textbooks would point this out in their story about Darwin's finches. So they will tell you that there's a 5% increase. See, that's an evidence for Darwinian evolution. But what they won't tell you is that when the drought ended, the beak sizes, they just oscillated about the normal. So there was no net evolutionary change from this experiment. So in fact, here is a statement from the National Academy of Science, which is saying that Darwin's finches are a particularly compelling example of speciation. Peter and Rosemary Grant have shown that every single year of drought can drive evolution change in the finches. So we have no new species originating here. That's the fundamental point. There was probably only the 15%, the birds that were remainder were probably just a frequency that has changed rather than anything significant. But what they've used this as an evidence, a particularly compelling evidence of speciation. And it's being claimed here that this is a compelling example of speciation because the grants have estimated that if droughts occur about every 10 years on the islands, a new species of finch might arise only in about 200 years. So this is heavily reliant on the extrapolation, which I showed you in this diagram here. So here you can see it's heavily reliant on the extrapolation. It's not the evidence. Evidence is just oscillating about normal. And there's no mention of the fact that the change was completely reversed in the following years, which should have been mentioned because that's what the evidence is pointing out. So Darwin's finches are a good evidence of natural selection because there was a 5% increase in the beak sizes for that small period of time during the drought. However, small reversible effects seen by the grants have been exaggerated to provide evidence of the origin of species, which it clearly doesn't. So what we really see here is merely small oscillations within species. And that's not an evidence for the origin of species that Darwin's theory actually requires. So here's one critic of Darwinism. His name is Philip Johnson. He's a law professor at Berkeley. And he writes that when our leading scientists have to resort to the sort of distortion that would land a stock promoter in jail, you know you are in trouble. So if stock promoters had done this, they would have been in prison. Yet we find leading scientists, evolutionary scientists, such as National Academy of Sciences, who work for this body known as National Academy of Sciences, have resorted to exaggerations of a 5% increase in beak size and making extrapolations from that evidence of a speciation event. So this is clearly exaggerating the evidence. And scientists ought to be more honest than stock promoters. That's what Philip Johnson's saying. Stock promoters would not be permitted to do that. They would not be permitted to exaggerate evidence like that, because if they did, they would be in jail. So scientists should be more honest than stock promoters. They are professional in their own respect. So they shouldn't be exaggerating small changes to macroevolutionary changes. So here is a summary. Although changes in size of Darwin's finches is a good example for microevolutionary change, extrapolations from the Grant's experiment does not compellingly support macroevolution, which is the evidence needed for the origin of new species. And we come to our last evidence, which I will discuss here, which is related to the four-winged fruit flies. And this is regarding the possibility of macroevolutionary change, whether it's possible or not, and whether there is any methodology that could be used in science to demonstrate that random mutations can produce a macroevolutionary change. And this is the third critique of natural selection. Natural selection requires variation, and it's the random mutations in neo-Darwinian sense. So creationists have been arguing that you cannot produce from random mutations beneficial macroevolutionary changes. So here I'll be discussing that. So here is a schematic illustration of what is needed for natural selection to work. So you have natural selection is three facts in a syllogistic inference. So you have fact number one: all organisms produce more offspring than possibly can survive to reproductive age. So that's overproduction. Fact number two is all organisms vary amongst themselves. And you can see that by looking at nature, and you will find that within each species, there is a slight variation. So this is a fact. And fact number three is that there is some principle of inheritance that operates in nature. In other words, at least some of the variation is inherited. So that's fact number two, the variation, some of this will be inherited to the next generation. So natural selection is three facts that we have just discussed in a syllogistic inference. So what is the source of variation, which is the raw material evolution? In neo-Darwinian sense, variation comes from random mutations, DNA mutations that are thought to be source of new variations and the raw material evolution. So now mutations do happen in nature, and that's a fact because sometimes the DNA replication has errors in it that leads to mutations. So this is a drawing of bacteria that's being targeted by an antibiotic. And this is the famous example where mutation is shown to be beneficial. Whether that's the case or not, I'll explain. So on the left side, what we have is antibiotic interacting with epitope on this target. It targets the bacteria. So when antibiotic binds to the epitope, it kills the bacteria. So that's how antibiotics work. And on the right side, you have a mutation that has been introduced in the target, which is the bacteria. So on the epitope of the target is mutations. So the antibiotic is not able to fit onto the target, which leads to a conference, which confers resistance to the bacteria. So this is called antibiotic resistance. So this sorts of beneficial outcomes at the molecular level have been found in nature, and these are beneficial outcomes to specified environments. So this is not a mutation in itself that is beneficial, it's a benefit with regards to a specific environmental change or environment. Mutations are never beneficial per se, they are beneficial with respect to environment. And here you have, remember another point, that mutations, they are only observed to decrease genetic information overall. So, there is a hypothesis in genetics known as antagonistic pleiotropic effects, where you have, through this, you can explain that there are some beneficial outcomes in specified environments, but overall the actual organism suffers harm. And there, if you want to read more about this concept, there is a book written by Professor John Sanford in 2005. It's called Genetic Entropy. And he proposed an idea known as devolution. We're going towards degenerative state. Darwinian evolution requires that we should increase in fitness. Professor John Sanford, he gave an antithesis to that, which is that we are not moving forward towards a fitter state, rather we are degenerating towards a less fitter state. So anyway, the point here is that mutations do happen, and here we have a mutation in the bacteria that confers it resistance to the antibiotic, so it's not removed from the system. So that type of mutation is specifically related to the beneficial outcomes rather than the mutation being beneficial itself. So here we have Darwin's theory, and it requires more than molecular changes, so more than mutations. Just to show mutations is not enough for Darwin's theory to be proven. It's not sufficient. Darwin's theory requires more than molecular changes. And the complete requirement of Darwin's theory would be to also show changes in shape and morphology of the organisms and life. Darwin's theory requires that as a necessary requirement. So it's not sufficient just to show molecular changes. So do we find mutations that can change morphological features? Neo-Darwinists, they present these type of examples. So these are two examples. Here's a mutation on the left known as antennapedia. A mutation in this gene leads to, antenna, a stubby antenna growing out of this region here. So you can hardly see them. And mutation in ultrabithorax leads to extra set of wings at the hull tiers, which is the balancer for the, so whilst the fruit fly is in flight, these are the balancers here. And can you see it's called hull tiers, the balancers. An extra set of wings grow because of mutation in 3 mutations in ultrabithorax gene. So remember that this mutation is a loss of function mutation. It's inactivating mutation. It doesn't increase genomic information that the neo-Darwinists need for the Darwinian evolution. It actually decreases. And these are called homeotic mutations in fruit flies, both of these. So that's why homeotic genes. So here we have the wild type normal fruit fly, which has a set of wings growing out of its normal region. And on the left side here, you've got antennapedia mutant, which has a pair of legs extended from the antenna, as you can see here clearly. And this is disabling. And on the right side, you have bithorax mutant. And here you have the extra set of wings growing out of its hull tiers, which is, again, whether that's beneficial trait or not, we'll discuss in the next few slides. So at first glance, what we have is the requirement of Darwin's theory, which is change in shape and morphology, being fulfilled through molecular changes. So we've got here in this in this example, you've got mutations that's leading to apparently beneficial traits, which is extra set of wings. This is obviously disabling, but this apparently appears to be beneficial, and neo-Darwinists will give you this example to show that, look, mutations do actually produce beneficial outcomes at the level of morphology and structure and shape. And that was required, so you can see from here. Well, several things to notice here. Firstly, this phenotype is cleverly designed and engineered in the laboratory by combining 3 mutations. So what we actually have is artificial product of genetic lab that is designed and not natural product. For Darwinian evolution, the product needs to be natural. Geneticists who are engineering this in the lab are intelligent beings, and if they are producing these mutations and putting them into specific locations on the genome, then that's an intelligence, that's an input by an intelligent agent, and that doesn't prove Darwinian evolution. So that's the first flaw in this line of reasoning from the Darwinian point of view. Secondly, the phenotype has been duplicated here. So we haven't had any new features that have arose. Darwinian evolution requires an evolution of features, i.e. slight modifications, and we don't have that here. We just have identical additional features, and that's not what Darwin's theory requires. We don't have any novel additions. And we got rid of the halteres, which is a problem. So that causes the fruit fly to lose its balancer, which during the flight, which is an impediment. Thirdly, the new phenotype is inert addition. So the new wings have no muscles, and they just sit there useless. They don't enable the fruit fly to fly any better than it was flying before. Rather, it makes the fruit fly have this extra weight on its back, which is unnecessary. So the conclusion is that this is a handicapped fruit fly. Neo-Darwinists will have you believe that this is a fruit fly that's got something beneficial to it. But the actual matter of fact is that here you have a handicapped fruit fly with all this extra useless weight on its back, and also a loss of the halteres, which is the balancer during the flight. So it's in its first appearance, it appeared to be a beneficial trait, and reasonably so. But if you really look at the detail here, this is not the type of morphological change that Darwinian evolution needs. And Darwinian evolution will not, natural selection will not select for this, because it's an addition that actually reduces fitness rather than increasing fitness. So it's an intermediate between one state to another state in a stepwise process, which is not increasing any fitness. So it's an evolutionary dead end. It cannot survive in nature. So I would argue here that as far as the evidence goes, genetic mutations are not raw material for evolutionary change, because firstly, a mutation has to be beneficial to be preserved by natural selection. And here, as you have seen in the fruit flies, the legs growing out of the antenna is disabling, and the extra set of wings growing out of its halteres is just creating extra weight on the back of fruit fly, which is of no benefit in terms of fitness. And secondly, evolution requires both change in morphology and molecules. And in this case of fruit flies, we saw molecular changes at DNA level, so mutations leading up to changes in the structure and anatomy of these fruit flies. However, the change was not beneficial, so it did not fulfill the complete requirement Darwinian theory needs for its evolutionary process to be fulfilled for the kind of claims that are made at the level of pattern of evolution. Thirdly,
There is currently no evidence for beneficial mutation that specifically and positively affect morphology. Several experiments are underway, but these experiments are only showing that the mutations lead to a reduction in the genomic information. They lead to switching off of the genes and so forth, along with a small amount of benefits that are beneficial with respect to a specific environment. So E. coli experiments being done in Michigan State University is a long-term evolutionary experiment, and that experiment actually shows that the E. coli that they are subjecting to the laboratory environment will not survive in its natural habitat. So it's not an increase in fitness, it's decrease in fitness. And wherever beneficial traits are yielded from such experiments, it is with respect to the specified laboratory conditions. So it's with respect to environment and not the mutation itself. Mutation was never beneficial in the first place. So here's a summary. Mutations in ultrabithorax are engineered to produce an extra set of wings that are disabling. And secondly, there are no known randomly occurring beneficial mutations that can explain the origin of myriad species that we see on Earth.